Hello everybody. How are we all doing today? So we are starting our chapter on hair analysis. So this is the first part of hair analysis. So let's have a little review. So who can remember what's the difference between class evidence and individual evidence? Take a minute to see what you remember. We just did this in the last chapter. Okay, so you should have remembered that class evidence would narrow down to a group of people but not a specific individual. So generally, hair is considered to be class evidence. However, if there's a follicle present with a blood supply, then DNA analysis can be done, and that would make it individual evidence. So hair is actually a good piece of evidence to study because it's easily left behind at a crime scene. It also resists decomposing. So if the hair stays in place, you can get it from a cold case even years later. And hair will also indicate if someone's been using drugs or was poisoned. And as long as the hair is still on the head, that drug use will show. So the reason why mammals have hair, and all of them do, temperature regulation to keep warm and protection, decreasing friction, and in some animals like cats, hair is also used as a sense organ. So here's a diagram of hair structure. The shaft is the part above the skin. It's composed of a protein called keratin. Consists of 3 layers from the outer to the inner, which we'll talk about later. The follicle is the part that's below the skin. This is the living part that contains the blood vessels. The cuticle is the outer transparent layer of the hair shaft. Okay, this is what looks sort of like shingles or scales, and the scales or shingles point toward the tip of the hair. So one of the reasons why people use conditioner is to keep the cuticle scales going down and keep the cuticle scales from being roughed up. And the cuticle scales laying flat are what allows the hair to look shiny. The cortex is the part beneath the cuticle. This is the biggest part, and this is the part that contains the pigment melanin, and that's responsible for the color of the hair. Melanin is also responsible for the color of your skin. The medulla is the center of the hair, and the medulla is classified into 5 different groups based on how it looks. And we can actually compare the hair shaft to a pencil. The cuticle would be the painted outside part. The cortex would be the wooden part that's beneath the paint. And the medulla would be what's commonly called pencil lead, but it's actually graphite, the stuff that you write with. So that would be your medulla. So here is a magnification of human cuticle scales. They're very flattened and narrow looking. These are known as imbricate. All humans have an imbricate cuticle. Animal hair or fur, we'll discuss later. Here's another picture of the imbricate cuticle. Okay, so the cortex, that's the biggest part. It's the main body of the hair. This is what contains pigment granules. You can see here, there's some air spaces here in this picture, and this has a little different pigment distribution here. So the pigment granules are really small granulated structures, and depending on the hair, the size, color, distribution is different. Okay, so here are your medulla patterns. So this is what they look like here. So you'll want to draw your diagram on the handout that I gave you. The continuous medulla is one unbroken line of color. The interrupted or intermittent pigmented line is broken at regular intervals. Fragmented or segmented, very similar to the interrupted, but the intervals are unevenly spaced. So sometimes you have to really look along the length of the hair to determine whether it's an interrupted or fragmented, because you may look and part of the hair looks like it's interrupted, but then as you look further down, you see that they're not completely evenly spaced. The solid is the pigmented area filling both the medulla and the cortex. And there are also people that don't have a medulla, so you don't see any separate pigmentation. A lot of times you see this with people with very light hair. In fact, when I first started looking at hair samples, I looked at my hair and my sister's hair and my niece's hair, and I was wondering why I couldn't find a medulla, and apparently none of us have one. But once I started looking at other samples of hair, I realized that the medulla was easy to find. You either have it or you don't. Okay, so here's a couple of pictures. Okay, so this is a hair with no medulla, so you don't see anything there in the center. Here is the interrupted medulla. Notice that it's pretty fairly spaced at regular intervals. This is a hair with a clear continuous medulla. So here's two pictures. See if you can identify the medulla pattern. So write down what you think. Okay, hopefully you looked at this and you realize that this is unevenly spaced. So this would be a fragmented or segmented. This has a pigmented area that fills both the cuticle and the cortex. This would be considered a solid medulla. So one of the things that first has to be done at a crime scene is to determine whether the hair is animal hair or human hair. So there are several differences to allow you to do that. So we're going to go through all three. And we can look at the pattern of pigmentation, the medullary index, which I'll explain momentarily, and the type of cuticle, because animals have different cuticles. Okay, so as far as pigmentation goes, humans have pigmentation denser towards the cuticle. In animals, it's denser towards the medulla. Animal pigments are found in solid masses called ovoid bodies. Human hairs are one color along the length, at least naturally. Color treating is a different story. Animal hair tends to have a banded pattern. So when I look at medullary index, it's the diameter of the hair's medulla, too fast on that, the diameter of the hair's medulla divided by the diameter of the hair. So if the medullary index is less than 0.33, the hair is human. If it's greater than 0.5, the hair is animal. So here we have cattle hair. Medullary index is 0.5 or more. Okay, the human hair medullary index is 0.33. Okay, so our cuticle types, the coronal or crown-like, generally found in rodents. The spinous or petal-like is found in cats, seals, and minks. Imbricate cuticle is found in humans. So here's a couple of different cuticle pictures. So looking at these, can you tell which ones are from an animal and which is from a human? Okay, so here's the mink hair. See, notice the difference. This is a spinous cuticle. It looks like petals. It's different than the way the imbricate looks more scaly. The bat hair, the coronal cuticle, look at these. They look like little crowns. And you can see the human hair. Here's a picture of cat hair. So we see a lot of ovoid bodies here. See these dense areas of pigment. Dog hair. Again, a lot of dense areas of pigment. Look at that large medulla. So you'd have a large medullary index. Rabbit hair. Deer hair. The deer is interesting because there's no cortex and there's a medulla that's just filled with all these little spheres. Okay, so sometimes at a crime scene you'll find hair from different body parts. So there's different textures and different characteristics that you'll see. Head hairs tend to have a soft texture, moderate diameter of the shaft. Pubic hairs have a coarse, wiry texture. They can be tapered, rounded, or the braided tips, and the shaft looks buckled. See that? Facial hairs tend to have a triangular cross-section and a coarser texture. Eyelash and eyebrow hairs are saber-like in appearance, meaning they come to a point, short and stubby, and they're usually curled. Limb hairs are also sort of curly. They have like an arc-like appearance. The tips are rounded and braided, and the scales are rounded due to wear from clothing. Okay, so that covers part 1.